And I'll admit people now.
Good morning, everybody. Welcome to our workshop on grouping and read across. We're due to start on the hour at 11 o'clock CET. We're just going to give people a few minutes because we still have some people entering the room. So we'll aim to actually start the presentations at 5 past the hour. So if people want to grab a last coffee, please feel free to do so. And then we'll start directly at 5 past the hour. Thank you. And again, for those who have sort of just joined, we're still getting people coming in. So we'll start the actual presentations at about 5 past the hour. So there's time if you want to quickly grab a last coffee or a last drink of water, and then we'll start in about 3 minutes. And again, to those who have just sort of joined us, welcome. We are just about to start at about 5 past the hour, just because we still have some people coming in. So please feel free to grab a last minute drink. We'll start in about 2 minutes with the presentations. Okay, so good morning, everybody who's on European time. Good afternoon, good evening if you're in Asia, and a very good morning if you're more in the US based. My name is Sean Kelly. I work for the Nanotechnology Industries Association, and it's my pleasure this morning to be the chair for this webinar. We will start with the talks. We have 3 talks being given this morning, and then we'll have an opportunity to have a panel discussion. The way we're suggesting this for the delegates is during the talks, if you have having any immediate questions, if you add them into the Q&A, and we can try and address one or two questions that's related to the talks. The question should really be focused upon a technical issue or a very specific issue. Anything that's more discussion, we can deal with in the panel discussion. During the panel discussion, there'll be an opportunity for people from the audience to also join in. You can either submit questions into the Q&A, or indeed, if you put your hand up, we'll be able to unmute you and bring you into the discussion. The agenda for this morning is we'll start with Kai Paul, who's going to give us an introduction to the ECHA framework around grouping and read-across approaches for nanomaterials. We'll then have Damiana Drobny from the Accord projects talking about their application towards grouping approaches on graphene and 2D related materials. And then we'll have Elise, who will join us from the iCARE project, talking about the application of grouping approaches on graphene and 2D related materials. For the panel discussion, we'll be joined by Virginia Rodriguez Umano from ECHA, from Stephen Hodge from Viserion, and by Vendel Vulleben from Bauer. And they'll be coming in to give some feedback from their different stakeholder perspectives, and also to start the discussion so we can talk about this whole area of sort of grouping and read-across approaches. If I turn over first to Kai and say, Kai, if you're ready to start, please feel free to do so. And for everyone else, please sit back, listen, and as I said, use the Q&A for any questions. And so over to you then, Kai. Thanks ever so much.
Yeah, thanks, Sean. Let me just try and this showing. So hopefully everyone can see that. So yeah, I'm Kai Paul. For people that don't know me, I am a principal regulatory consultant at Blue Frog Scientific, mainly working within the EU and nanomaterials. Like Sean said, I'm going to give you a brief introduction to an ECHA project that finished last year, at the end of last year. And there were a couple goals there, which I'll explain later on. But the main focus here, considering what the workshop is really to focus in on the identification of single nanoforms, which is grouping, but not really grouping, and some of the state of the science of the group for grouping of nanomaterials that we saw as we went through this project. So before I really start, obviously it wasn't just me on this project. There's a list of some of the people that were involved, and some of these people have previously been involved in some key projects such as Gracious, the read across framework there, and Sunshine, and lots of other things where it comes to Similarity and grouping for nanomaterials. So I'm going to give a brief project outline. I'm going to go over some of the state of the science that we saw so far as grouping and read across Similarity. I'm going to discuss sameness of nanoforms under REACH. I'm then going to go into the concept of acceptable variation and then mention some of the project progress and then on to the conclusions. So before I dive into the project outline, I think one of the most critical things that we sort of learn on the way, and one of the most critical things to bear in mind as I go through this, is that the way that the REACH legislation is written for nanoforms, each one of the nanoforms is considered to be different a priori. That means as soon as you have a nanoform and you create another one, it doesn't matter how close it is or how similar you think it might be, it's considered different. What this led to is some people may be familiar with one substance, one registration. That's where you consider that you all have something, I don't know, like formaldehyde, and you get together and you say that you can just share some data, and therefore you have one substance, one registration, one data set, and that minimizes the amount of animals and effort that it takes to register. By making nanoforms a priori different, it becomes incredibly difficult for that process. In fact, almost non-existent because of the way that it's written into the legislation. Of course, expanding on that concept, you have one substance, one assessment, which then bridges across the different regulations within the EU. Now you can imagine that if one substance one registration or OSAR is difficult, then one substance one assessment also becomes even further difficult, especially when there's lots of differences between those regulations, including things like the definition of a nanoform or a nanomaterial. So what's harder for nanoforms? Sameness. So our first goal was to try and develop and create boundary compositions or a way to make boundary compositions that would be inclusive of the nanoform characteristics which aren't currently contained in that. So traditionally, when you have a substance, you would base it on the composition. So do I have formaldehyde? What are the different impurities in it? Are they similar or similar enough to other people? So, and these were really based on pragmatic cutoffs. So for a monoconstituent, there's the 80-20 rule, where it has to be 80% of one particular substance, and then you can have 20% of varying impurities. Those kind of cutoffs simply don't exist for nanoforms. So that was really our goal, is can we create these cutoffs which lead to some acceptable variation, which then lead to the same profile for a nanoform? What do I mean by profile? Is it the same across the ecotoxicity, toxicity, and physicochemical endpoints and data requirements? The second goal of the framework was the project was to create a framework for sets of similar nanoforms. Now, sets of similar nanoforms were put in place to try and reduce the strain of compliance for nanoforms, because obviously within the regulation, it was written in that every nanoform would be different, then the sharing of data technically wouldn't be allowed. So what are the other processes that we can do to ensure that there is a reduction in animal testing? And from that born was sets of similar nanoforms, where you have to prove that your nanoforms are similar enough that the then data would also be similar, so the profile in itself would be similar. But there is a lack of a framework at the moment for industry to follow on those requirements to prove the sets. And then off the back of that, there's also a lack of a framework for the regulators to assess against. And what that's led to in the last sort of several years since the nanoforms came in 2018, well, 2020 officially, is that there's been huge divergence in how people put these sets of nanoforms together. And then there also then isn't a uniform way to assess these, which further causes confusion as well between the different stakeholders. Because of the uncertainty in and around it, the data intensiveness as well to prove these sets of similar nanoforms has actually been quite burdensome, but it is incredibly unclear on how to do it, and it's open to interpretation. So during the project, what we wanted to do is look to see if we could find any rules and to piece together or any cut-off criteria that would then help towards this kind of assessment. Why are these two things and two goals really important? I think at the moment, the uncertainty of things around sameness and sets of nanoforms, it leads to a less ideal marketplace, and sometimes an off-putting marketplace for people that perhaps want to get access to the EU. And subsequently, we can constrict the innovation of key enabling technology. So I'm going to start by going over some of the state of the science. We had varying search strings, and from those search strings that included grouping and sameness and all the different endpoints that we'd be interested in so far as reach, we found over 1,500 manuscripts. We reviewed them, and after a review, less than 150 we considered appropriate, relevant, and reliable. And of those 150, not all of them were grouping, but some of them were perhaps on QSARs and other concepts that we could use in grouping frameworks. And if you look at this graph here, what you can see here is the type of studies or the type of endpoints that were mentioned in those different manuscripts. And I think the pattern's probably quite obvious to everyone what we've ended up with. There's lots that mention size, There's lots that mention that dissolution, lots that mention shape, some more on reactivity, and then less on things like attachment efficiency, which are incredibly important for how things move through the environment. If we break this down even further, what we can also see is from these manuscripts, what percentage did we have on each kind of study type? And you can see that it was dominated by physical chemical endpoints. Now you see I've put Annex 6 next to one and Annex 7 next to another. That's just the REACH annexes. Annex 6 is things like size and shape, crystallinity, for example, and then Annex 7 are things like flammability or log KOW, which we know isn't relevant for nanoforms anyway. And you can see it's well over 50%, moving into 70% for that. And then a lower level for in vitro, and then around 17%, even less for in vivo studies, which are critical studies that you need towards REACH, which then becomes critical when you're building a framework, and even less again for environmental fate. And if we look at the endpoint categorization, we see that it's hugely skewed as well towards toxicity. But just the presence of these manuscripts, of course, doesn't mean that they're usable. And also, in some ways, things that we've studied can be quite limited. So there's quite a lot on fiber toxicology with good cause, because of course we all knew asbestos, and with some of the fibers and things that people were creating, we wanted to make sure that that was or was not something that was going to happen with them. So I said, a lot of the sameness, similarity, and grouping approaches were focused with good cause in and around things like shape and size, because these things we know can change the absorption, the desorption, the excretion, etc., where it's going to go, how it's going to act, how reactive it is. But there are many aspects that are covered to a lesser degree that can, of course, then impact the profile of the nanoform. And here's a nice graphic that courtesy of Vicky Stone, which shows some of those things and then what it can impact essentially. So the charge of the nanoform and the molecular will impact the molecular interactions, for instance. When we look explicitly at the Annex 6 information and we look at what was the most covered to the least covered, size and shape was quite dominant, followed by surface area, followed by things like coatings, crystallinity. And interestingly, the concentration of the constituents or the impurities or the additives sometimes weren't even mentioned at all. Now, I think from a manuscript writer's perspective is like, of course, that's going to change things. But when we don't know from a regulatory perspective, it becomes very difficult for interpretation if we don't know the exact percentages or what kind of impurities are there. And also critically, from an interpretation perspective, if we don't know the impurities, we don't know if it's those that's driving the actual characteristics of the nanoform. So, for example, out of 10 studies that we saw using zinc oxide nanoparticles, out of those, there was only two, I think, that reported, one or two that reported the additives and the impurities. Other endpoints used for grouping probably won't surprise people: reactive oxygen species, size and shape, surface reactivity, dissolution, attachment efficiency, the biodegradation of organic surface treatment of chemicals as well from an environmental perspective. But when we're looking at these and we're thinking about trying to create a framework for REACH, we've got, we have discrete and fairly non-discrete studies. So we have some studies that are very much yes, no. So you look at a screening study for genotox, that's a very much a yes or a no study onto whether it's genotoxic, certainly from an in vitro perspective. But then we have much less discrete studies that have lots of different endpoints, lots of clinical observations, like reproductive studies. So although these grouping frameworks are based on things like reactive oxygen species, can we actually make that leap from a reproductive study or a neurodevelopmental study? And the answer is simply we can't. We can't make that leap, and that's one of the things that makes it incredibly difficult when we're trying to go from, some more of the in vitro studies to this hugely to a complicated in vivo study. Another thing that further compounds some of this assessment is we're still missing some test guidelines. For instance, some of the in vitro tests. Yes, one of the skin sensitization tests is indeed validated now, but we actually need the suite of three to cover the whole adverse outcome pathway. There is a lack of guidance on what is needed for sets of similar nanoforms. Dissolution is still missing, even though we know how critical it is so far as test guidelines. And also the interpretation and type of test for fulfilling things like size can significantly change the result. And to the point during our research, when we were looking through this, we were looking at zinc, and this was one of the graphs that we got from the information from the different types of zinc. And you can see there, I think it's M113, the orders of magnitude difference in the size that was reported. Now, this could be method differences, batch differences, etc. And that can really start to skew any analysis or when you're trying to build a framework on cutoffs or rules. So yeah, like I said, I mean, things like dissolution, things like size, we know are going to drive the eco-toxicological processes, but there is still uncertainty, and because of that uncertainty and the lack of test guidelines, we still have divergence, and it means that comparison is very difficult. The dosimetry and the measurands people use can be different as well, which can again makes any comparisons difficult. And there's often poor reporting of nanoform characteristics. And that just, further widens that gap between certainty and uncertainty. So when it came to sets of similar nanoforms, during the project, I mean, what we saw is that at the moment, it wasn't possible to, with confidence, to allow to build this framework on some solid sort of rules and cutoffs to lead to extrapolation across all reach data requirements. That they would be substantiated or not support the higher tier tests, and they would be focused on a small set of nanomaterials, the metal oxides, the monoconstituents, and as well, I guess, they wouldn't necessarily, the rules based on the literature search that we had done, it wouldn't protect all targets sufficiently, so environmental toxicity wasn't well covered, for instance. And obviously the inconsistency of methods. And that simply, I'm not saying that it can't be done, it's the time frame that we had within the project wouldn't allow their development. So yeah, here's a couple more things that I think I've already touched on, so I'm going to go back over them. And again, just to say, like, in future, we certainly can build some guidance, some assessment guidance for sets of nanoforms, but it would not be solely reliant on rules and cutoffs. So we shifted the focus to sameness, which was part of this project anyway, but is it sameness? Well, at the start I said every nanoform is different, so therefore they can't be the same. Under REACH, sameness is a well-defined substance, and if they consist of the same constituents, that determines their sameness. So if we don't have sameness for nanoforms, in fact, what we're looking at is there acceptable variation between the characteristics of a nanoform, or a framework where we can use it to identify nanoform boundary compositions towards joint registrations. So why have we targeted this if I've said that there's loads of uncertainty and that we couldn't possibly do it? Well, the first step is, okay, how simple can we make it, and what's the first step in the regulation? Well, the first step in the regulation is what substance do you have, and is it similar to anyone else's traditionally? By reducing the variability, if we say that things are the same or acceptable variation where everything else has to be the same. If we reduce that variability, it also inherently reduces the uncertainty. And just because we have a lack of data thing doesn't mean that some of the fundamentals are gone. So inter and intra-laboratory variation stays the same. Batch-to-batch variation will stay the same for people, and biological facts like size for absorption and translocation. So for instance, we know that about 5 nanometers isn't going to necessarily cross the skin. So it's these integrations of some of the biological knowns on top of some of the regulatory knowns where we decided to focus. So things like CLP cutoffs, so classification cutoffs, is also something that we can build into the framework. So on the right-hand side here, you can see how you would decide what your substance is. Does this work for nanoforms? Absolutely does. It's not broken. We can take that forward. But the usual next steps would be to reach out and see if anyone else has the same substance, which we can do from a compositional perspective. However, what we can't do is it doesn't include the nanoform characteristics, which we know can have an impact on the nanoform profile. That's not the same for something that goes into solution. So therefore, jumping straight to testing doesn't necessarily work. Now, the nanoform requirements are defined again under Annex 6. So what we wanted to do is try and find a way to integrate this, and we call it the acceptable variation rather than sameness because that doesn't exist for nanoforms. But what we're trying to do from a reach perspective is build a substance identity profile, and that is essentially a group of people that can get together. They decide on the boundaries of the nanoform characteristics as well as the composition, and then they can share data together. So we're looking to build what we've mentioned as a nano SIP. When you fall out of this nano SIP, you move on to the sets of similar nanoforms. So what we did for that is try to base it around a very simple decision tree framework for each Annex 6 nanoform characteristic, as well as composition. And the work that we've done for that will be published, I think, possibly this week. I can't mention too much on it right now. I will give an example of a decision tree. But I thought for context of this workshop, it would be good to give some background on state of the science and where we're still lacking. And to mention that, 2D nanoforms also weren't very well covered, which means some of this work is really critical. But yes, it will be published this week. And here is an example of what the decision trees look like. So you can see from the start, it's okay, is it less than a 3:1 aspect ratio? And then we can say it's a spheroidal type nanoform. Are they the same size? Where we used a 1.5 times difference, which was actually built upon the ECHA Nano app. And a lot of the foundation was built along the ECHA Nano app. And then some of these critical biological cutoffs. Is it less than 10 nanometers because things start to change there? Are they all less than 5 nanometers or greater than 5 nanometers because that's skin penetration, and then 3 nanometers for things like crossing the blood-brain barrier. And you move through these decision trees and you basically have to answer yes to everything to say that you have the same nanoform to be able to create data just all based on the Annex 6 information to fulfill the rest of the reach obligations. So I'm going to wrap it up there. Like I say, it will be published. I will be presenting at the NanoMaterial expert group meeting at the end of this week also, and there will be further dissemination on it after that. But what we saw is robust approaches for sets can still become data generation and animal more cost intensive. The uncertainty for sets is actually in that way no different from read across or the difficulties in defining QSARs, where you have to build on layers of evidence to get to complex endpoints. There is limited data at the moment to base it on and to validate the grouping on, and that as well is skewed, as you've seen from the beginning of the talk. So we focused on the creating of boundaries for single nanoforms, because by creating these nanosips, we can move the regulation back into one substance, one registration. There's a lack of information on 2D and platelet materials, and I think that's where projects like iCARE and iCARE have both come in and have started to further address this and create data. There's also a lack of ways to go from in vitro to these more complicated endpoints like reproduction. And again, Accord and iCARE are trying to address this in different ways by having these, adverse outcome pathway or mechanistic approaches. And iCARE will present later about how they're doing it, grouping for neurotoxicity. And data for registration and cost of nanoforms is high, and that really the project was just trying to increase the proportionality between what you do for a nanoform and what you do for a normal substance. So why must we focus on here from a regulatory perspective? To get a workable regulatory procedures and frameworks, to ensure safety of humans in the environment, to increase market accessibility and confidence and clarity, which encourages business and innovation, increases confidence in what is required. That then subsequently increases the data pool. By increasing the data pool, we can have more data to pull on, and increases then the certainty, and we just end up with this continual feedback loop where we can improve the frameworks and the regulations off the back of it. And one other thing that I hear quite often is, well, we can learn a lot from what we've put in place from nanomaterials for advanced materials or polymer reach. And at the moment, we haven't solved this one. So I think it's critical as well that we keep working on this. And with that, I will say thank you very much for listening. And if there's any questions for clarification just now, I think that's fine. But I think main questions will be later on. And please do feel free to get in touch.
Thank you ever so much for that, Kai. We've not actually had any questions, but in the Q&A as yet. So hopefully we can keep all the questions then to the panel discussion. I will move swiftly on and we will welcome our next speaker, who is Damiana, and she's going to be talking about the Accord's application of grouping approaches on graphene and 2D related materials. Thank you, Damiana.
Thank you very much for a nice introduction. Do you see my presentation or not yet?
We do.
Okay, thank you very, very much. Yes, now I'm going to present the contribution of Accord's project to this topic, Sameness and Similarity. So this is Accord's team. We are altogether 10 partners. The project is coordinated by University of Ljubljana. And the vision of the Accord's project is we are trying to explore the full potential of imaging-based techniques that were underused in the past. And we are focusing on 2D materials. We have taken graphene and graphene oxide as representative materials. And we want to at the end, provide something like a safety profile. So we can even name it differently, but it is all about linking material characteristics to the safety. So our concept is as follows. So we are still working, finalizing on graphene family materials. We are working on materials synthesized under very well-controlled laboratory conditions and also from materials from the company. And we have pre-selected a list of methods, a group of methods for physical and chemical characterization, and also a group of methods for biological or hazard identification. So the idea was that we have selected or put together the methods different partners have expertise on. So therefore we call it a pre-selection, and the aim was to select methods with the most applicable to the materials. And then the idea is for different groups of materials to select first methods and then to elaborate the safety profiling. So this is the structure of the project, just for a very quick introduction. So laboratory synthesized material synthesized by the industry, then development of this physicochemical biological characterization, and we want to bring as much as possible our methods toward the standardization. So the pathway to some impactful outputs is as follows. So we selected our methods on the high throughput, medium throughput, and low throughput methods. And we would like to make a list of methods that are applicable for characterization of 2D materials at the place of synthesis, in the industrial settings, then in the expert lab and in a competence center. So we know that methods have different, also different costs. Therefore, they are not applicable everywhere and for all different purposes. So this is a list just for your information, what we have selected for the physical chemical testing. This is a list of methods that are already used, and we thought, or we already had experiences in the past, what made work for 2D materials. And here is even a larger list for the biological or hazard testings. So we used a lot of high throughput methods and then medium throughput methods, low throughput methods, organoids, and so, and also try to link the characteristic to some environmental and human health hazard. Just for your information, we have characterized in detail more than 50 laboratory synthesized materials, where also the synthesis process is very well known. So we can link the synthesis with the characteristic of a material. So we can really go deep into as safe as possible by design. And then we have characterized as much as possible also industrial samples. Why as much as possible? Because sometimes it is just not possible to characterize this material that is already incorporated in matrices. So with all the methods. So this is a big list of materials we have worked on during these years. Now I'm going to very quickly show why imaging is so very important and why we are quite happy with the final outcomes, because we were able really to use label-free imaging techniques to see what we really detail what we were working with. So here we see very nicely with scanning electron microscopy these 2D materials. So when seeing in detail, you can really get an idea of what you have, what material you have, and not just that, you can get really this idea of variety of different shapes, sizes, layers, and so on. And here we see a crosscut of a cell in vitro that internalized this material, the very same material. And what we see inside cells, that there's something going on with the material inside cells. So partly degraded, partly changed, and these cells are usually not really affected. This is just a normal process of internalization, probably also exocytosis then, and so on. So we can really see what is going on with a cell. We can then, based on these, also do or select some further tests. For the purpose of grouping, we selected these mathematical models. We use correlation analysis, then principal component analysis, and partial least square regression analysis, just for your information, to perhaps better understand, or just to get an feedback on what was behind the data we have also produced. So here we can see the correlation between different material descriptors, and we have taken biological molecule absorption as a measure of reactivity of a material. And what we can see, what we have seen, is that there's no single variable that perfectly correlates with absorption, with characteristic of a material. What was actually expected, because we know that there's not just one characteristic, but many of them. And we were also interested, is it possible, would it be possible to group materials according to their characteristics? And we did first some preliminary testing. We have taken 6 different materials, graphene, graphene oxide, and MXenes. And based on 6 materials only, we could not group them in groups. Yes, so they seem to be different materials, similar but different. But when we have taken more than 50 materials, coarse materials, we can very nicely group them because we have more materials, the model is more accurate. And in this case, based on the list of different characteristics, they look same. We find this as a very important finding. So when we have enough data, there are models that could very nicely show us whether we have same or similar material. What were the characteristics that were most relevant or the most important for the absorption of molecules, so for the reactivity? As expected, interlayer spacing. So it means the more spaces in between layers, the more reactive material is, the more the material absorbs molecules. With other words, the more surfaces are available. And then on the other side, peak disorder means the material is somehow messy. There's not that much surfaces, and this characteristic was just the opposite. In this case, material was not that reactive. So again, we can really confirm that there's no single variable that dominates this absorption, but they act jointly. Some contribute more, some others less. And here we see, I'm not going to go into detail because we are going to sooner or later publish it, which of the characteristics are most responsible for material absorption. What we see here we have more materials. Here we have materials from the main study, not just from the preliminary. And based on the characteristics we had and on the absorption strength or potential or intensity, we can very nicely group graphene and graphene oxide as same group. So all graphenes, although they have very different characteristics, and all graphene oxides, they have very same type of behavior. And we also link the cytotoxicity to the material characteristics, to the material. And we also see here that graphene and graphene oxide group as different groups. So all graphenes as one group and all graphene oxides as the other group. So they behave as two groups, but within groups they're not differences. So it means that also with this cytotoxicity test, they could be considered as same group. So we could talk about sameness. When we look at the reach requirement and the reach information for nanomaterials, which is shape, aspect ratio, particle size, distribution, crystallinity, surface area, and surface treatment, and then translate these into our characteristics, we get this. So when it comes to shape, the best methods that perform best are these: XRD and then electron microscopy. We cannot talk about shape in case of 2D materials. We talk about number of layers, also size of stacks, perhaps also visualization of particles. So we try to describe the, not in terms of size distribution, but perhaps more in some size of flakes or something. So we don't know which descriptors the best describe the shape of the material. So it's not just a shape, but terms are a little bit different or adopted to 2D materials. And this shape contributes, has a medium contribution to the absorption of molecules. Then lateral size dimension, XRD is the method of choice. Again, we talk about number of layers, again, medium contribution. Then aspect ratio, it is very difficult to talk about aspect ratio in case of 2D materials, perhaps crystallite size or size of stacks. It depends, or it's a matter of a discussion in the future. Low contribution. Then particle size distribution. We talk about agglomeration size of agglomerates. has medium to high contribution to reactivity. Then thickness, it's also difficult to talk about thickness. Perhaps the best would be number of layers, size of stacks. About chemical analysis, we in our case, in case of iCARE, so we talk about carbon-based materials in all cases, but when we compare this to vaccines, of course, that's a little bit different chemical composition. Also, in our case, we had some functional groups, but they are not included in this model yet. And the highest contribution to reactivity was the surface area, what we translate or adopt to the material as interlayer spacing. What I have mentioned, density, we think it's not applicable. And crystallinity and crystal structure, we think the best in 2D materials would be defects. We are not sure yet. It's a matter of a discussion, finalizing the project and all the results. If we look at this table, what we can see that when we take into consideration all the characteristics that are included in rich required information, we can see here a list of methods that could best address them. Of course, XRD, electron microscopy, UVVs, and also Raman are those that could best be used and applicable for this purpose. Then we also see that in case of 2D materials, we can somehow translate or adopt these characteristics also to 2D materials. This is for sure not the final, let's say, shape or naming. It's a matter of a discussion among our project and also among EU dealing with regulation, so we can adopt this, but for sure this could be also used for 2D materials. So what is also outcome of the accords? So we also recommend methods, because we said we would like to recommend methods that could be used on the spot in industry and also in different labs. So the simplest, the cheapest is also UVV. So this could be very nicely used for this first characterization approach. Then XRD is also very useful. And of course, imaging, electron microscopy, perhaps electron microscopy, SEM, scanning transmission mode, and then EDX is a very useful equipment that is already present, very widely present in different industries, also in different labs. So could provide a very nice information for further decision making. So we already have a list of methods widely available and could be explored. So the regulation could also include the requirements that are based on these methods and could not be that difficult to provide. So what are actually the conclusions so far? We don't have, these are not the very final conclusions, but they will for sure not change much. Data are, of course, very important. We know that. And in a way, we have worked, not just our project, but also iCARE and others. We collected data in a way to be shared as fair as possible, of course, and in a way to be that could be processed by available mathematical models. So PCA or PLS approaches are very appropriate to identify relationships. And when we identify relationships, then we can also identify or make grouping. So the second important conclusion is that graphene and graphene oxides are two distinct groups, similar. But when it comes to biological reactivity, to biological characteristic, to safety, they could be treated as same. We find this as a very important information. So we did it with the cytotoxicity assays. It was very important in iCARE that all these assays were label-free. We have selected or tested a lot of available cytotoxicity tests, but in case of 2D materials, with materials that have color, they're dark, there's a lot of interference. So we have at least 2-3 methods that are label-free and reliable. And also by using these methods, we can confirm which materials are safe, are same or similar. So what we think it's also important finding is that which required information is appropriate also for 2D materials, of course, with some adoption, and of course with some additional characterization protocols, and probably also some standardization toward naming and terminology. What we think we already have enough knowledge and expertise that this is not something what would be too difficult for the past, for the future, and that in 2D materials, again, surface area is very important. Particle size distribution, no matter how we will call it, or in case of 2D materials, and of course defects. They have most important role for biological entities, for absorption of molecules, and for interaction with cells. And of course, all this is very relevant also for the safety assessment. And this safety assessment goes into the direction of assessing safety profile and of course for the application. I think this is all from my side for now. I'm very happy to be included or answer questions then at the end of the meeting. Thank you.
Thank you ever so much, Damiana. I don't think I'm not seeing any questions in the Q&A, so we'll move straight on to our final speaker, who is Elise from Temasol. She will be talking about the work being done in iCARE around grouping. I'll hand straight over to Elise and allow her to talk, so we allow plenty of time for the panel discussion afterwards. Thank you.
Oh, thank you, Sean. Sorry, I will keep my camera off during the talk because the sound system is not always good with my computer and Teams. Sorry for that. So I'm I'm happy to be here to discuss approaches with quite a large audience of, I think, expert in the topic. I'm working at a mass solution, and within iCARE, we work with graphene 2D materials and neurotoxicity endpoints. The target is to really establish techniques and methodologies that can evaluate nanomaterials and composites in relation to neuro-nanotoxicity. Focus on graphene 2D related materials and composites with particles from 3 industrial partners. So Avanzare, Advanced Material Development, and Verscion. And we added what we expected to be graphene data material from the GRC, the graphene GRC. So in total, we are playing with 6 graphene, 1 graphene oxide, 1 oxides, and industrial partners also generated composite samples representative of release particles during the use and the end of life of 5 nano-enabled products. So it's one of the aspects that I will not go into, but that is covered in iCARE as well. So currently we are integrating results from multiple research institutes with physical-chemical characterization techniques that I think in our core previous presentation, were well discussed about. And here I will focus more on the abiotic in vitro and assays that partners are developing within the project that will be used to support grouping and read-across approaches. So the target is to develop an integrated approach to testing and assessment for neurotoxicity and select a minimal panel of cost-efficient assays that could predict neuro-nanotoxic effects of graphene 2D related materials using read-across and grouping and read-across approaches. When we think about toxicity, there are 2 really important parameters to consider to make something developments relevant for real life. of course, the exposure potential and the biopersistence, because to create neurotoxicity, studies showed on the particles smaller than 2 micrometer, that they can affect the brain through olfactory tract and blood vessels via the respiratory systems, and create neurovascular dysfunction and damage and inflammation that can lead to neurotoxicity. But these particles should persist and be accumulated in some ways. So repeated inhalation exposure are usually required. In the case of pristine graphene materials, we expect that those materials, if inhalated, have a limited biodegradation from literature again, tend to persist and aggregate inside biological media with, of course, some interaction with proteins and dispersants. So, today, is the application of existing frameworks, the grouping approaches developed in Gracious and the ECHA read-across assessment framework, which are really using a structural similarity as a heart of the frameworks. It's kind of a prerequisite and a rich, to have same composition material, so that's key elements. You need to really deeply characterize your materials, which I mentioned it could be quite demanding already in terms of cost and time and some aspects if you want to go with a full characterization as presented earlier. Of course, you need to understand the use and exposure scenario, as we spoke a bit earlier, with a repeated inhalation, potential for persistency, and have behavior related reactivity data to be able to start forming grouping hypotheses, so forming sets of nanoforms. And I mentioned them, but there are already some thresholds developed for nano descriptors inside the ECHA NanoMap. Those can be used initially and to create this grouping hypothesis. And as we work really on the right side of the scheme with more developing integrated approach to testing and assessment with, again, a minimal set of assays to be able to do this refinement of hypothesis. So, as it was mentioned earlier, we need multiple descriptors for graphene related to the material identification. There are hundreds of more or less distinct metals that can be classified under 4 categories: the graphene, graphene oxide, reduced graphene oxide, and functionalized graphene. And within those categories, we have potential for material variability, and there are some different other uncertainty aspects that can be linked to the measurements variability. And as for most of the nanomaterials, they are quite subject to extrinsic properties transformation. But if we stick to primary descriptors, intrinsic descriptor, we can have already some potential grouping consideration regarding those 4 categories and a bit within the categories, the subcategories. So as mentioned, there are some key physical-chemical parameters that seems to be really important, for example, the layer number in the case of 2D graphene-related materials, the defect density, the interface chemistry, among others. And so there are already quite accepted ratios. If you go with a platelet, you need to have a ratio between the lateral dimension, the width and thickness, that is a 3-fold higher than in pairwise comparison, with the thickness for the lateral dimension and the width. We can have some subgroups with we have monolayer, bilayers, fluolayers, graphene, but also graphene with a bit more numbers, as it's the case in our project. And some considerations that may needs to be validated with data in terms of like interlayer spacing and such characteristics. Yeah, for the moment, like we are still in the process to incorporate the data from the different partners, which is quite time consuming. But if we do really basic Euclidean distance assessments on some of the materials that we have data kind of rich, we can see that already like we have two sets of nanoforms be formed when you take into account qualitative DLS data, because not applicable quantitatively for graphene, but it was like several experiments in different mediums that were used to be categorized and input in there. The layer count from the TM data, defects density, and layer structure from Raman ratios and XRD. So we can see we have already some clustering with some available thresholds that Kai mentioned as well from the ECHA nano. Like we can already also apply just like on one by one characteristics some fold change analysis to do a grouping. That will lead to, if you take into account those display characteristics, leading to GNP1, Coke, and forming a group, same than here actually, and AVAN1 and GRC forming a group as well. So as an example, but like the ECHA nano app, of course, there are some elements that may need to be like carbon-based, specific, and even more 2D. But there are those dimension of tiers. So first you go with structural similarity that we spoke about now, and after you can allow a kind of higher fold differences in those characteristics if you complete your analysis with further testing. And so there, like there is this in vitro toxicity aspect where you can consider like fold changes as well. As Kai mentioned, like there is not that much structural activity relationships that have been developed quantitatively for graphene 2D related materials. We have trends that are reported in the literature, like if you have graphene oxides or reduced graphene oxides, you will have oxidative stress related endpoints that could be relevant. In the case of large flex cellular exposure, like you can expect a bit of membrane damage or due to sharp edges, for example. But the descriptor-driven nano-QSAR for 2D graphene nanomaterials are really limited and usually limited to absorption and interaction studies. And there is not kind of like QSAR OECD compliant existing. And that is due to the high uncertainty, due to lab-specific small and heterogeneous data sets. That is really limiting any extrapolation. I think in iCARE you have quite a good number of materials to really start speaking about categories and maybe trends definition. But in the case of iCARE, we have 8 materials, so it's a bit limited. The graphene properties are subject to strong environmental transformation, and there are other kind of consideration, applicability of ecotoxicity assays for these materials that lead to, even if data indeed sometimes they are not that reliable. And so there is this approach which go a bit outside of the structural similarity, but taking into account the bioactivity similarity, where the new approach methodologies are really key to support the grouping and substantiating the similarity. So you can predict neurotoxicity effects in advanced model using more simple models, for example. So this kind of hypothesis that we will like to look for inside the data set that we are currently consolidated. And so the quantitative activity relationship may be possible to draw. So we have like a high throughput phenotypic profiling, high throughput screening, high content screening, and omic technologies provided in the iCARE project. So it will be kind of like extensive different level to integrate. And we are targeting, as I mentioned earlier, this as close as possible AOP IATA approach. But just to give a bit of example, so we have iCARE that have developed, for example, 2D co-culture human models of brains with neural cells and microglia. Those are evaluated in high throughput imaging assays with different staining that highlight different cellular compartments, generating like a really important amount of data to consider. And we have other dimensions, like the high content analysis with genotoxicity assessments, DNA damage. So I apologize for the ChatGPT creation, but it was really better looking than my slide initially, so sorry for that. So we have like a kind of several ROS abiotic assays in the project, and after a panoply of cellular to tissue and more organism level assays. Some are already getting out of the game because of not compatible with the graphene materials and the composite samples. And so we are currently reviewing all of those data coming from different models. So for example, for the BBB, so the blood-brain barrier models, we have different settings to represent it. So this cross-tier data integration and weight of evidence will be used for to support identification, grouping, and read across, providing hopefully mechanistic information. In the iCARE, the objectives, and I hope Steve will be able to comment on that, will be to really help doing a fast and cost-efficient decision and screening priority for further assessments. We are in the context where we expect more and more safety demonstration from products alongside their life cycle. So maybe this kind of type of thing could be relevant, knowing that the neurotoxicity is not really validated. There is no validated approach, even lots of effort toward it for in vitro assay battery. And so like maybe the generated information, like the FISCAM and the neurotoxicity multi-output will be relevant also for regulatory purposes. So we are ongoing. So I'm afraid I don't have that much conclusion because the target of the analysis is really to go do a good data pre-processing, transformation, and analysis workflow to get reliable and reproducible grouping and cross outputs. And because there is a variety of algorithms, models, and tools, and we are reaching the end of the project this year. So now that the data are coming along, so we are starting to test. So again, as for basic statistical analysis, but we just in including some of the cure data from iCARE partners, we can see that, for example, the previous grouping where we got like the GFC and the amendment that were like grouping together now is not reliable anymore based on PCA. Again, we need to be careful here, and there's plenty of for complementary analysis to do. But those kind of data will help refining the grouping analysis, and iCARE is working on developing an in silico pipeline for data consolidation. So using like templates, raw data templates, and going to the potential read across assessment with visualization modules and things like that. So that is under the work of INL partners. So Kabir started working on that. And so I hope to be able to share more, maybe in a follow-up events on the finding of the project. Thank you very much.
Okay, thank you ever so much, Elise, and thank you to all of the speakers. We do have a few questions in the chat, but we can come to those in a minute when we start the panel discussion, and we should do that straight away. So I'll just quickly introduce the panel and allow people the opportunity to see who's on the panel. So, as well as our three speakers that we've already heard from this morning, we're going to be joined by Stephen Hodge from Viserion PLC, who is one of the industrial partners in the iCARE project, and he's very much coming in with more of an industrial perspective. We're going to be joined by Virginia as well from ECHA, who's going to really be looking at things from a more regulatory perspective, and Vendel, who is working at Bauer, who has also probably got more of a regulatory perspective, but also in his previous life in BSAF, was one of the people that also helped develop the ECHA Nano app that people have talked about. So I'll just quickly set the scene by asking each of the new panel members a quick question, and then please feel free if people have other questions to add them into the Q&A, and then we can address them. So Virginia, sort of having heard about the fact that there's a lot of different assays that can evaluate the behavior or reactivity of materials, and that this is something that's really important about influencing the grouping outcomes, and especially thinking that carbon materials sometimes, or 2D materials, are sometimes challenges. I really want to sort of say, are there any sort of assays that are being recommended by ECHA to justify read across, especially for carbon and 2D materials? And how would ECHA expect to see some of the data being presented by somebody submitting a dossier that would allow ECHA to make a decision on the sort of grouping approach?
Yeah, hello, good morning. Thank you for the opportunity to be here. So regarding your question, ECHA doesn't have a explicit list of recommendations when it comes to this grouping of read across. So we don't prescribe any specific testing. So basically the approach that remains is this weight of evidence or hypothesis driven approach. So in order to do that, so what we recommend is that to the registrants that the explicit grouping hypotheses that they have been used, the justification of the descriptors that were used for the assays are well documented. Also that maybe to help the comparative data matrices, and then to link the physicochemical properties to the hazard outcomes to start with. So basically, and to sum up, when applying for a read across or a grouping approaches, what we are expecting is that the studies or the endpoint for which they are applying, they have adequate and reliable coverage based on the test that they are using or for the specific endpoint.
Okay, thank you ever so much for that, Virginia. Turning to Vendel next, if I may. Vendel, obviously in your kind of previous life, you were one of the key developers of the ECHA Nano app, which has obviously been mentioned in the talks today. Really the question is, do you think that either the Nano app or other sort of similar methods that could support grouping and read across could be adopted or adapted to actually manage graphene or other carbon or 2D materials?
In principle, yes. I mean, what I saw today from Elise was a creative way, and I think it's the right strategy to focus on one endpoint and then, like Virginia also said, create evidence to perform a weight of evidence approach. And of course, we have to stick to what we have. We have the regulatory text Annex 6, which tells us exactly what we know, and this is what we have to take as a basis.